Hello students, welcome to my channel Nurturing Intellect. Today under chapter Biotechnology Principles and Processes, we are going to study the vectors which we use for plants and animals. So let's begin.
So students, we have seen in earlier class that plasmids and bacteriophages, they are the vectors which we use for bacteria. Okay. Now, if we need to transform plants and animal cell, we can't use these plasmids or bacteriophages because plasmids are found only in bacteria and bacteriophages are the viruses which infect only the bacterial cell. So we need some vector which will help us to deliver the foreign gene in plant cell as well as animal cell. Okay. So the vectors which we use, they are genetically modified pathogens. These are the pathogens which cause infection in plants and animals. So we disarm them, we remove the pathogenic gene and insert the gene of interest in them so that they can transfect plant and animal cell and deliver the desired gene. For example, in case of plants, we make use of bacteria, Agrobacterium tumefaciens. Okay, so this is Agrobacterium tumefaciens, and for animal cell, we make use of disarmed retroviruses. Let's study Agrobacterium first. Agrobacterium is known as natural genetic engineer because it has the tendency to transfer a portion of its gene into the plant cell when it causes infection. This bacteria lives in the soil and it causes infection in many broad-leaved crops, that is dicot plants like tomato, soybean, sunflower and cotton, but not in cereals. Okay, so as it causes infection in the plant cell, okay, the plant cell start producing tumors. They start dividing at an uncontrolled rate and form tumors. These tumors, as you can see, they are known as crown galls. So how it is transferring its genetic material to the plant cell? So here you can see this is an Agrobacterium tumefaciens. It has its bacterial genome. In addition to that, it is having a natural plasmid. The plasmid is named as TI plasmid. Okay. A segment of DNA of this plasmid called tDNA. It transfers when it infects the plant root cells, it transfers this tDNA. It has the natural tendency to transfer this tDNA into the plant cell. Okay, here you can see. And that tDNA, this tDNA, it goes and integrate with the chromosome present in the nucleus of plant cell. So it becomes the part of the chromosomal DNA of plant cell. And this is coding for repeated cell division. This gene is coding for repeated cell division. So such plant cells in which the tDNA gets integrated, they start dividing at an uncontrolled rate and forms the tumor. The tumors are called crown galls. So as the tDNA causes tumor, that's why the plasmid is named as TI plasmid. And since humans are not at all involved in this type of transfer of gene, that's why Agrobacterium are known as natural genetic engineer. See the procedure in detail. So Agrobacterium, this is Agrobacterium, chromosomal DNA, and this is the TI plasmid. So it transfers its tDNA naturally when it comes in contact with the plant cell, okay, especially the wounded plant cells, okay. So it transfers its tDNA to the plant cell and that tDNA goes and integrate here, okay, in the chromosome. So now this whatever the gene is coding, it will start producing those proteins. Here you can see the structure of TI plasmid. This portion of the TI plasmid is known as tDNA, okay, where there is gene for auxin, cytokinin, opine. So these are the plant growth promoters. So they causes excessive growth and causes the formation of crown galls in plants. Understood why Agrobacterium tumefaciens is known as Mother Nature's natural genetic engineer? Now, scientists, the geneticists, they manipulated this property of Agrobacterium tumefaciens
And they used TI plasmid as vector for plant cells.
for introducing a foreign gene into plant cells. So let's study the procedure. First of all, we take out the TI plasmid from the bacteria Agrobacterium tumefaciens. And how we are doing it? By using lysing enzyme. Okay, so we treat Agrobacterium tumefaciens with lysing enzyme, lysozyme, that breaks the cell wall, and then we treat with chemical that breaks the cell membrane, and we isolate TI plasmid. Similarly, we isolate the DNA from a foreign cell. Now we treat both TI plasmid and foreign DNA with the same restriction enzyme, and by adding ligase we obtain recombinant TI plasmid. So when we treat with restriction enzyme, TI plasmid with restriction enzyme, this tDNA we remove, okay, and in place of that the foreign gene gets inserted. So this recombinant TI plasmid, okay, it is used to reinfect a new agrobacterium. Okay, so the transformation occurs, the agrobacterium will pick up the recombinant TI plasmid. In the next step, we have to do the selection of recombinants over non-recombinants. So for this, when we were introducing foreign DNA, along with that, we integrated an antibiotic resistant gene, that is kanamycin resistant gene. So those agrobacteriums which had picked up the recombinant TI plasmid, when you grow them in a medium containing antibiotic kanamycin, only the recombinants will survive and non-recombinants, they will die. These recombinant agrobacterium cells, they are used to infect the plant cell grown in a culture medium. The plant cells which are used here are meristems of the host plant. Okay, so as it has the tendency, the TI plasmid has the tendency to transfer the tDNA. So this newly introduced DNA, it gets transferred into the plant cell and goes and integrate with the genome of the plant cell. Now through tissue culture technique from these cells, we can develop several new plants. So all the cells of this newly generated plant will have that foreign gene, and that gene will express itself to show a new trait. So this is how we are making use of TI plasmid for creating genetically modified plant or transgenic plant. In case of animal cell, we make use of retroviruses as the vector. So retroviruses, they have RNA genome and they have reverse transcriptase with them. Okay? So they have the ability to transform normal cells into cancerous cells. So as they infect the animal cells, they become cancerous and they start dividing at an uncontrolled rate. So once we remove the toxic gene, the cancer causing gene from the retrovirus, this process is called disarming the retrovirus, we can introduce a desirable gene in that position and it can be used to transfect the animal cell. So in this way, the foreign gene gets delivered in the animal cell and it will express itself. So from all this, what we can conclude, that once a gene or any DNA fragment, we ligate it with a suitable vector. We introduce a foreign gene into a suitable vector. That vector can transfer that foreign gene into bacteria, plant or animal host cell. And after getting transferred, it starts multiplying inside these host cell. So with this, we completed the topic cloning vector. Using next class, we will study host with methods we use to transform competent host with recombinant DNA. You must have liked the video. If you like the video, in order to grow my channel, please hit the like button, comment and subscribe to my channel. Thank you very much.